everybody, welcome to Skype a Scientist Live. Today we are talking all about viruses with Efra, and so we're super excited to do this. And here's some stuff that you should know. Tomorrow we're going to be talking about sharks. That's going to be happening at 1 p.m. Eastern. The following week we're going to be talking all about how to work at a museum, the kind of work that's happening at the Natural History Museum in Los Angeles. Also this week, we're going to be talking about outer space on Thursday and microbes on Friday at 5 p.m. Eastern. This is a non-profit organization that helps support these sessions. And so if you can help support us at all, even a little bit really helps. You can do that at patreon.com/SkypeAScientist or paypal.me/SkypeAScientist. So at this point, I'm going to hand it over to Efra, and he can introduce himself, and then we'll get into all of your questions. All right, take it away.
All right, thank you. So I am Efra. I've been studying viruses for about, 8 years or so. I started my journey studying bacteria fermentation back in undergrad. Then I went on to studying plants for a while, did my master's studying plants. And then I switched to studying viruses for my PhD, and I've been doing that for quite a bit. I did my PhD studying virus-host interactions and how viruses interact with the cells that they infect and how the cells try to fight back the infection. I then did a postdoc studying hepatitis A and how hepatitis A virus enters liver cells and codes and releases its genome and how it establishes infection in the liver. And then I did a second postdoc studying another group of viruses that includes hepatitis C virus and Zika virus, trying to find proteins in our cells that the viruses need in order for them to replicate. So all my training has been in that realm of that interface between what happens when a virus encounters its target cell and all that back and forth between the two. And because viruses are obligate intercellular parasites, which means that they can only replicate inside the cell. Outside of the cell, they are inert. They really cannot do anything. Essentially, all the steps in the quote-unquote life cycle of a virus happens inside the cell. So I use a lot of microscopy approaches in order to visualize what happens as soon as a virus enters the cell, and how it tries to make that cell its home while it establishes the infection. So that has been my focus for all these years, and it continues to be.
Awesome. So we're going to ask you a whole bunch of questions that are coming from all over the place. And so if you don't know the answer, that's okay. No shame. Sometimes we ask questions, some of which are reasonable and in your wheelhouse, and sometimes are totally out of left field, but we figure we'll give them a shot anyway. And so the first question we have from Leah is, what exactly does the coronavirus do?
So just like any other virus, I am now, when I'm starting to study respiratory viruses. I've studied cardiac viruses in the past and liver tropic, but essentially you have to step back and think of what a virus wants to do to begin with, right? So their sole purpose or what we would say a successful virus, what is the goal of a successful virus? And it's to propagate from host to host, right? If you just stay in one host, you can't spread. So a characteristic of essentially any virus is that it wants to do just that. So you can have different routes of infection. So you can imagine that a virus that transmits through the fecal-oral route is going to perhaps cause gastroenteritis and vomiting diarrhea, so that promotes shedding to a new host. In the case of a respiratory virus, it wants to do exactly the same. But in this case, it has to induce coughing and has to induce things that will allow the virus to jump from host to host. And that is a common trait for a lot of viruses, with some exceptions. Of course, in biology, there's exceptions in everything. But in this case, you have a virus that gets through the respiratory tract, and now we're starting to see in the literature a lot of back and forth. Initially, we thought to be only in the lower respiratory tract, which means just the deep part of the lungs, where air exchange happens in the alveoli. Now there's also evidence that there's replication in the throat, which is a part of the upper respiratory tract. But in essence, what a lot of viruses do, and again, I refuse to use the word all because there's so many exceptions in biology, but many viruses end up killing the cell that they infect. So you have a virus, the virus has to find its target cell, and a lot of viruses are very, very specific as to the type of cell that they infect. Otherwise, imagine if a virus was more promiscuous, the same virus will be infecting essentially every organism, every organ in our body. So they're very specific. In this case of the coronavirus, it targets very specific respiratory cells, as you can imagine. It has to then penetrate the cell, release its genetic material. So any virus out there is composed of mainly two things: is the genetic material that has the recipe to make more viruses, and that genetic material is protected by a protein coat called a capsid. So all viruses have that. Some of the viruses have an extra layer of lipids, fats, called the envelope. Some have two shells, but for the most part, they have protein shell inside. And inside you have genetic material. And that is why I describe as a genie lamp, if you will. So you have the lamp protecting the genetic material. And once it's inside the cell, then that genetic material gets released, being like the genie that will make more copies of themselves. So the coronavirus is no exception. It has to find the target cell that it wants, that will provide all of the things that it needs in order to create more copies of itself. The lamp opens up, you have genetic material, and you're going to have what we call, depending on the viruses, the order may differ, but you're going to have production of the viral proteins coming from that genetic material that will then replicate genetic material and more copies of itself. All those proteins are going to come together to form a new shell. And during that process, and once you have that going on, you essentially turn the infected cell into a factory. It's going to be producing thousands of different viruses. And although there's some viruses that do not kill cells, for example, some viruses that establish latency, for example, there's some viruses that kill a cell. You can imagine coronavirus is one of those. And by doing so, it causes damage to our bodies, right? You're losing your own cells. But a lot of viruses use this strategy of destroying our own cells. Imagine if you now have thousands of little progeny virus, what we call, inside a balloon. You want to like blow it apart so that now each one of those progeny virus goes and finds a new target cell, and the whole cycle continues. So from a disease point of view, there's a lot of things that trigger what we define as symptoms, right? You have from the fact that you have viruses using the machinery that we need to survive, right? We have cells because each one of our cells is doing something to keep us alive. So you have cell loss from there. But at the same time, there's a lot of responses that happen during a viral infection, and that is what I study. And I'll jump into there in a second. But also the body is responding to a viral infection in a very specific manner. So you can have, for example, inflammation. You can have cytokine production, which are molecules that our cells secrete to tell other cells what's going on. So you can have infiltration of cells that would normally not be there. So a lot of immune cells, B cells, C cells. So you can have a whole myriad of things that are going on. Some of them induced by the virus, such as it's destroying your cell, and some of them are in fact caused by our own body in trying to repair that. But in doing so, everything is a balance, so you're going to have symptoms that we have, such as lethargy and fever and all that. But going back to the first part when I stopped, and it's because I studied that for my PhD, and is that for the coronavirus right now, what we're seeing is a lot of the topics that have been discussed is the virus enters, it infects, it kills cells, and then the output, do you have antibody production? And because we tend to correlate antibodies and immunity to understand disease. And to me, that's always like the first point and the end point. You have infection going on, and what is the output? Somehow you develop immunity. But for the longest time, my main interest is what happens within hours of infection. And there's a lot that goes on, and there's a lot of lab studying this. So I'll just give you a little bit of an example. when a virus infects a cell, um, of course, the cells in our body do not want to be infected by any means. Viruses have been roaming around in this world for millions of years. So it's natural to imagine that our cells have also evolved ways to counteract viruses. So as soon as a virus enters a cell, our cell responds by activating like sensing mechanisms. So if you will, you can imagine this as an alarm system in your house. And there's someone trying to break in, and the first thing that happens is your alarm is going on. So cells, almost every cell in our body has a response for this. Um, and the whole purpose of this is to the final output of that, and that happens within hours of an infection. And the whole purpose is to alert all the other cells that have yet to be infected to let them know that there's a virus going on and that they have to be prepared. Unfortunately, also a lot of the signals that happen when this is activated and all these cells are now, okay, there's a virus going on, we need to do something about it. A lot of the responses overlap with, for example, inflammatory responses. So you're gonna have a lot of symptoms too. So a question as such as what does the coronavirus do to our body? A lot of things. Some of them because the virus is doing it, some of them because our body is reacting to it.
Awesome. Okay, so our next question is, when did you decide to become a scientist? And I think maybe you should speak up just a little bit because some folks are having a little bit of trouble hearing.
All right, better. So probably in undergrad. So as a kid, I wanted to be a veterinarian, and but I was born and raised in Puerto Rico, and we don't have a vet school in the island, so you have to move to the mainland or go abroad. And I never, my thought was always to just stay in the island. Little did I know, I ended up doing a PhD in a vet school, so fun things happen. But then I went to college and I didn't know what I wanted to do. I knew that I loved biology and I loved life. So there was a point where I was thinking of medical school because that's what we all hear about. And you think of that is the only route as a biology student. But unfortunately, I met a lot of people, and back then there was no social media like what it is right now. But I did meet a lot of people who helped me along the way into visualizing and exploring other opportunities. So I did that, and I started doing research in bacteria for a while. I did not like growing bacteria; it was too smelly for me. So I ended up just ditching it. But it was a fun opportunity to learn and to appreciate the power that comes with research, which is creativity, and just every day is different, and I like that about it. Then I went back to, so that I did a summer internship, and then I went back to Puerto Rico, and I took a course in botany, and I just fell in love with plants. It's, for many of the same reasons why I love viruses. They're essentially sessile, they don't move, yet they can do so much. And so I fell in love with plants, and I studied them for a while. And it, that probably was the moment when I said, okay, I can dedicate my whole life to studying something that not a lot of people care about yet. I just think that is fascinating. They don't move, they just, but they respond and they do all these different things. And so I did that. I studied plants for a while, and then I went into studying viruses, and that's a long story. But yeah, I think it was just that switch of me realizing that medical school was not the path for me, having a lot of connections and people with the right advisors at the right time that know you by who you are and your personality and what position will be the better fit for your personality, and also just being open, just being flexible to ideas that you had never thought about.
Awesome. We've got a next question from a couple people. Are there any viruses that can be good?
Yeah, so definitely. So I can give you two examples. I guess so. Just before answering that question, we're infected by a ton of viruses. Viruses are everywhere. They infect essentially any living organism, from fungi to amoebas to fish to every single organism that that is alive can be and is probably infected with a virus at any given time. And we're no exception. There's a lot of viruses, like herpes viruses, that at some point, essentially all of us are infected with it. And so if you think, for example, as what we call bacteriophages, which are the viruses that infect bacteria, and they're normally in our gut too, just like what we have a microbiome. Some of them are good because they're destroying a certain population of the bacteria in our gut so that they don't overpopulate, for example. And so that's one way that I'll consider them good. And people are using those phages or bacteria infecting viruses as a therapy. And so probably many of you are aware with antimicrobial resistance and how these bacteria are becoming resistant to drugs. So now people are exploring the use of these phages to go and destroy bacteria. And also a lot of people don't know that some viruses are very specific to killing transformed cells or cancer cells. So it's another realm of what we call virotherapy, of how we're using viruses for a benefit. And same applies in other realms. Also, plant viruses are some that we know that are beneficial to help plants grow. So definitely.
Awesome. Let's see. How is the coronavirus different from other viruses? That's from Katie, age 9.
Depends on what other viruses mean, right? There's a lot of viruses just come in every single flavor. If you can think about any different permutation in a puzzle, viruses can probably do that. They vary a lot coming from genetic materials. So some viruses have DNA genomes, some of them have RNA genomes, some of them are single-stranded, some of them are linear, some of them are circular. Like if you can imagine every single thing out there that you can change in a puzzle, there's always an exception for a virus. So again, I will just step back so that you can just imagine that a question like what is the difference you can take it from many different angles. You can take it from how successful is the virus at spreading. And if you consider that, you have to consider all the extrinsic factors that come with it. How stable is in the environment? How successful is the virus at finding its target cell? You can imagine that a virus that has to infect a nerve cell, if you just inhale it, probably will not get there. So you have to have that level. Species specificity, like how specific is that viral infecting? So there's a lot of things. For my sort of specialty and what I care about, how successful is the virus at shutting down the immune system? And that is what we call in our field as like any, like my advisor used to have a word like almost like any successful virus, so it's a word that it was kind of funny. But essentially any virus that will cause disease has to have a way of shutting down your immune system. And again, I'm talking about way before antibody production and way before adaptive immunity takes place. Because any virus that will want to cause symptoms, or in this case, a coronavirus causing a disease, has to find a way to shut down all the alarm systems in our body to sort of give it time for it to actually be spreading. So when it comes to differences, a ton of differences. There's a lot of papers now coming out about how comparing, for example, how coronavirus and other respiratory viruses, think of influenza, respiratory syncytial virus, how do they differ in the target cell they're infecting, how they respond or successful they are at shutting down the immune system? Can they modulate our own cells in blocking antiviral production? So our cells naturally produce antivirals as a sensing mechanism. Many viruses have evolved a way to shut it down. And now there's evidence that coronavirus can do that, and it's not surprising, a lot of viruses do, but that is also promoting the inflammation route, which in your case, you're going to have tissue damage, again, different from some other viruses. And you're going to have essentially every different single difference that you can imagine, it can happen. From my experience, for example, when I was studying cardiac viruses, you can have viruses that destroy the muscle cells in our heart. And if you compare that with a virus that infects the enterocytes that are lining your gut epithelia, those cells in your gut, they're very easy to replenish. You lose them, and there's another one because you have a stem cell niche. You don't have that in the heart. In fact, the muscle cells in your heart that you have right now are the ones that you had as a kid, because cardiac muscle cells rarely ever undergo mitosis. So if you have a virus now that is destroying your muscle cells that cannot be replenishable, of course, you're going to have a different condition than if you have a virus that is infecting a cell, such as a fibroblast, that can divide. So all of these differences come together into different manifestations of what we call disease.
Great, thank you. Gianna would like to know, I've heard that there are 2 antibiotics that have killed COVID-19. Is that true?
I am not familiar with what exactly.
I think is that the hydroquinone or something?
So there's a whole hydroxychloroquine and the other antibacterial. That antibacterial the azithromycin is what we take if we're going to travel to certain parts of certain countries to stop travel diarrhea. So that is azithromycin. It's using most of the times that when we take it is for that. And hydroxychloroquine is typically used as a malaria treatment. None of them are antiviral. So just like when you have an antibiotic, which will be specific to bacteria, then you have antifungal for fungal infection. You have antivirals. And in this case, you have hydroxychloroquine, which is an anti-malaria treatment. But what it actually does is that it inhibits parts in our cells and changes pHs, and just malaria and some viruses require specific pHs in the endosomes in our cells. So if you start messing up with that, you can prevent certain steps in the replication cycle. So this drug, even though it's used for malaria, for example, the way that it does, or its mechanism of action, one of the mechanisms of action is that by changing the pH in the endosomes, which are specific vesicles in our cells that transport different things. A lot of viruses, so once the virus finds a cell, enters, that lamp has to come apart to release the gene, right? Some viruses, this happens within the endosome vesicles. So if you start messing up with the pH, then that gene can never come out, so you don't have medication. So a lot of these viruses or a lot of these drugs are not specific to a specific virus, because what they're doing is modulating our own cells. In terms of the evidence for these two, I still do not personally believe it, just because we just don't know enough. And a lot of reports right now, you're gonna see this back and forth, yes and no, yes and no. And it's because of, unfortunately, we are living in a situation right now where we're just desperate for data, for what is going on. Can we fix this immediately? And by doing that, we're also having a lot of differences in how studies are done and are performed. And that comes from not randomized trials, or comes from very cherry picking specific populations. And you can imagine that changing things such as gender and age and status of condition and whether that person had a previous immune condition, like there's a lot of factors that if you're not normalizing for that, it's just impossible right now for us to say for sure these are working or not. But in the same realm, before we move on to the next question, I also want to make that difference that when we say antivirals, there's essentially, you can divide them into 2 different realms. You can divide them into drugs that are somehow doing something to our cells that the virus needs. So if you take it away, it just renders them and hampers them in a way that they cannot continue the replication cycle. But they are really altering something in our cells. While you also have what we call direct antivirals, which are then drugs that are really specific to binding or doing something to proteins or components of the virus itself, which you can imagine that will be better because they're not affecting our cells. Those are, of course, trickier to make, and it comes at a price that those are going to be or have to be specific for each virus, because a gazillion different viruses out there. The benefit of having one that would inhibit sort of a general step in our cell is that you can repurpose it for different viruses as long as they use the same machinery, the same drug. And that is the whole premise of why people were using hydroxychloroquine, because this virus uses a route that many other viruses use. So if it works for this virus, perhaps it can work for this one. Versus using something like remdesivir that it's a drug that directly binds to the viral protein, in this case, the protein that makes more copies of the viral genome, then that is more specific. But by doing so, of course, that doesn't mean, or that means that you cannot just repurpose it to use it for any other virus out there.
Great, thank you. Michelle would like to know, can viruses infect other viruses?
So yes and no. So I think back when I was taking virology, I guess the thought is they can sort of say it like yes, but I will say more of as a codependence. So there's definitely examples of viruses that for some reason their genome is, imagine, half as what they need. So if you get infected with this virus, it just cannot do anything on its own. So there's different viruses that rely on a secondary infection to provide all the components that is missing. And once that happens, and you can imagine the ratio or the probability of a cell in an organism to be co-infected at the same time with the two different things that happens in order to produce the output, it's minimal, but there are different cases of that. So in that case, I would not call it as a parasite or a parasite of the other. It's just really rely on the other. But you can see how the interplay can take place.
Cool, I didn't know that. Vito would like to know, are you optimistic about COVID vaccines?
You want the personal opinion, or do you want
just like, sure, your personal, emotional approach to this.
No, I definitely, of course, before a scientist, I am a human, right? And I'm a family person, so I also have my hopes and expectations. I don't know. And I don't know in terms of, vaccines just take regularly months, years to develop, right? And we're here in a rush. And thankfully, as the name implies, right, SARS coronavirus 2, there was a number one. So there's a lot of information that we can extrapolate from the first time. And that is why you get a lot of publications are just coming out every day. You just refresh bioRxiv for preprints, or even journals like Cell, for example. I go there almost every 3 days, and there's a new paper coming out. And a lot of people are going to think like, why this didn't happen for this virus, for example? And it's because we have a lot of precedents. We have other members of the family, and that we know a lot about. So something that you'll see, for example, is that when we're talking about organisms and you have like genus, species, right, and they're italized, and you're like, yeah, this is a genus and this is a species, even though viruses we don't write their names like that, they are taxonomically classified, just like family and genus or species and whatever. And because of that, there's a reason for that. There's a reason why they have a family and they have all this, and it's because they share a lot of characteristics. And that is why it's so important what we call bench science or basic sciences, because without that, right now we'll be in an even worse position. And it's because of studying other viruses. In terms of a vaccine, so for SARS, coronavirus 1, the one that we got before, it just sort of disappeared. I think that's really the term that people are using now. When people ask like, what happened to the first one, it just disappeared. So we don't know. We never got to the point where like it is needed right now. There's different platforms. A lot of different groups are working in different platforms. But making a vaccine is not as simple as one can imagine. And there's a lot of things that we can do in a lab, for example, in culture of cells, that does not necessarily mean that that is what you're going to get in an organism that is composed of thousands of cells, of many different cells, and you don't know how one thing can affect the other and the whole cascade. So there is that, and just like any other, again, taking a bacteria, for example, in terms of antimicrobial resistance, you don't know how the virus will evolve. You don't know, there's a lot of different things. So I want to remain hopeful. I think we're moving as fast as we can, thankfully, because we have so much background from other viruses in this family, but I don't know.
All right, great. Let's see. So a lot of people are asking, like, if my brother gets coronavirus, will I get coronavirus? If my mom gets it, will I get it? Like, is there any kind of genetic likelihood? Like, if you're related to someone, are you more likely to both get it or both not get it?
Yeah, so again, yes or no. So the first part of the question, yeah, there's definitely whole genetic factors involved in any condition, right? And that's why you're going to have some genetic based conditions because they're in our genome. And infectious disease is not an exemption. One example that a lot of people don't know is that, again, virus needs to find its cell and enter. It does, though, by typically binding to a receptor. And so in the case, and that's why ACE2, you're going to see a lot of in the literature for coronavirus too. In the case of HIV, it has its own receptors, which are different. And maybe people don't know that if you have a mutation in the co-receptor for HIV, you cannot get infected. So, and this is just based purely on genetics. So in this case, even if your partner has HIV, you can never get infected. You're naturally resistant to infection. So first part of the question, yeah, definitely. We don't know what those host factors are. Or at least I'm not aware of any specific mutation or suppression or whatever you want to call it that will render you completely resistant to an infection, but it can certainly happen. You can imagine that it can happen from the receptor side, also from how our bodies naturally fight infection. So there's different what we call polymorphism, which are changes in our DNA. That's why we're all different. That's why I'm a little more brown than Sarah, for example. So there's different polymorphism, for example, in immune genes that encode different proteins that protect our body. So if you have different polymorphisms, you can react differently. But if you just normalize by that and you start removing all those little permutations, then really what you're going to end up with is what is the chance of transmission, number of contacts and distance. If person number one coughs or is talking, what's the probability of those droplets that are coming out of your nose or your mouth to carry infectious particle, not just or infectious virions, not just the virus, for example. What's the probability of those to get into your right part of your body, in this case your mouth, your nose, to then get into your throat and establish an infection. So any virus out there has to fight all these different layers to get to where it has to be. It's not just as simple as you touch it and it's infected. So you have to always take into consideration all this. I don't know if that really answers the question, but it's sort of like you have, in order for you to answer any question regarding to how this is the output of this, you have to then consider all the little things that have to happen in between.
Yeah.
And I think when this all exploded, and I was getting calls from newspapers and all that to explain, for example, like hand washing or hand sanitizer, I realized that a lot of people, including my mom, who lives with me, non-scientist, she has her little sanitizers here, and she's always sanitizing. And that's really when it hit on me that in her mind, and I'm pretty sure it's common for many people, they sort of think like the viruses just come and naturally appear from the environment. And so even for example, my mom never leaves the house. So I had to explain to her, if I don't leave the house and you don't leave the house, yeah, I mean, there's always a chance, right? But you don't have to be sanitizing your hands every day or every single hour while you're in your bedroom. It's not sporadically going to emerge. So getting back to the question, it all comes down to probability of contact and frequency, and it's what sort of, to end the question, is sort of what we call the Swiss cheese model, and is that in order for A and B to happen, if you want to get to point B, imagine a Swiss cheese, and every single thing that that needs to happen has to go through each hole. So if you switch or move a slice of cheese around, then that's going to be blocked. So meaning there's a lot of different things that have to happen in order for that hole to, in that cheese block, to go perfectly, so that you have a successful infection.
Awesome. Next question. Why can cats get coronaviruses, but dogs cannot?
Yeah, so that was published, I think, last week. It was a preprint for a while, and then it got published. I actually don't remember the rationale. So of course they did first, they did a whole comparison of different domesticated animals and also farm animals. So they included pigs and chickens and ducks in the study. And they also included ferrets, because ferrets are a really good model for respiratory transmission. And by transmission, I mean, in a mouse example, in a mouse experiment, for example, we do what we call an inoculation, which means you literally take the mouse and you inoculate it with a virus, see what happens. Does it cause disease? You have, what is the cause of disease? Can you block it with this antiviral? But mice do not sneeze, so you cannot really use mice for transmission experiment, meaning if you infect these mice, and not this one, but you just put them in adjacent cages, how can you transmit from here to here? But ferrets do sneeze, and they have been used a lot in the influenza field to test this, to see dynamics of transmission. So this paper did see it in ferrets, you can have that. But surprisingly, in cats too, there were some correlations in terms of, it appears that younger cats tend to be more susceptible. They had some data suggesting also that it can be transmitted from cat to cat through the air. Similar principle, you can imagine cat here, and you have another case with a cat here. Infect one, can get to the other one. But I also want to, and sort of like, that is a descriptive, right? You have, it can happen if you follow these steps. And by that, I mean, of course, the first cat had to be inoculated. In this case, was intranasally. So you just take a cat and you put it in the nose. You also have to think, every time that these studies explode, you have to think, like, what is the probability of this happening in your house? You're not injecting, or you're not making a cat inhale a virus, right? So a lot of these things can happen. So in this case, yeah, it seems like cats can get infected. Last one I saw, I think, was yesterday. There was a preprint sort of trying to get at this, why, and then why is this happening from, for example, the receptor side. Is it that the receptor that the virus uses to enter the cell is close, the cat one is closer to, so you have, and it's just not sequence-based, you have to look at a 3D model, and it's the right cell expressed there. We know what makes ferrets more susceptible or not for influenza, because it has been studied for so long, but when it comes to cats, it's just too recent to pinpoint something.
All right, awesome. The next question is, are there any animals that are completely not affected by viruses at all?
I hate saying none in biology, but I will say probably in this case, no. I think at the very least, there's going to be some retrovirus genome, and retroviruses are viruses just like HIV that the way that they work is that their genome gets integrated into our own genome, into our chromosomes. Um, and there's many viruses that, so that viruses can do that, or they can just remain latent by other means. Mean that you're not necessarily like shedding viruses at 100% of the time, but those sequences are there. And that I will argue that every single organism out there can be or was or is infected, whether it is an active infection or not. I guarantee that it is present.
Awesome. We can always say like none that we know of yet, and then that hedges our bets. We always gonna have I was talking to one of my friends who's not a scientist, and she was like, "You always have these little like asterisks."
Yeah, it depends.
Well, the biology is so expansive. How could we possibly know definitively? Because there's so many exceptions.
Yeah.
Okay, so we still have a lot of questions open, but we try to end these sessions at 45 minutes, and we're already at 42. And so we just have two more questions to ask you, and then maybe people can find you online if they're still super curious, or maybe you can give some suggestions of people, maybe things they can read or something. But your final two questions are, one, what is something that you wish everybody in the world knew about your area of expertise? And the second question is, what is something that you wish everybody in the world knew about literally anything? It can be as simple or like significant as you'd like.
All right. Oh, those are really good questions. I'll respond to the first one with what baffled me when I started studying viruses, my very first semester of virology, and it was the notion that because a virus is essentially genetic material coded by protein, but those proteins are encoded by the genetic material, you can essentially make a virus completely synthetic. And that is always my argument when it's the whole debate of whether viruses are alive or not. But there are bacteria that are obligate intracellular parasites. Why are they alive and viruses not? And I'm on the side that they're not, and it's because of the whole notion of, in the case, for example, of coronavirus, that has what we call an RNA genome. So if we go back to molecular biology or the central dogma, our cells have DNA, and the DNA then encodes RNA. The RNA encodes for protein. There are viruses that their genome is DNA, just like ours. Herpes viruses, for example, papilloma viruses. Coronaviruses have an RNA genome, so they're already here. Meaning, if you just take the RNA and you introduce it into cells in culture, you can get virus out. You don't need an infection and starting infection. And if you know the sequence, like now we have the sequences of coronaviruses, it's out there. It baffled me and just wowed me that all I had to do is go to a company, and just like the way that we design small sequences of DNA to do polymer exchange reaction or any other assay, you can actually synthesize the whole sequence of a virus, make RNA out of it in a test tube, put it in a cell, and the cell will produce virus because you gave it the recipe. And to me, that is still this day, I'm like, it is just so fascinating that you can just create infectious particle from something that you can just make. It's just a sequence of nucleotides. So I wish a lot of people would know that, and to me, that's fascinating. In common, doesn't have to be virus. That is a good one. I think it has to do with more mitochondrial transmission of, I think I would love for everyone to know and to realize that our cells as eukaryotic organisms, we have DNA in our nucleus, but also in our mitochondria. And I would like for people to appreciate the role of female inheritance and the DNA that is in mitochondria, which are these organelles that produce energy in our cells, which are also very important for fighting disease. And that is a whole new topic. It will take me about 45 minutes. But because mitochondria are really important to fighting viral infections. But there's DNA there. And in the case of humans, I would like for people to appreciate that during reproduction, the mitochondrial genes that we get are strictly from, or largely, not all, largely from our mothers. So.
Awesome. Is there anything you'd like to plug?
None of this. No.
Okay. Where can we find you online?
I am on Twitter almost all the time at naked capsid, and capsid, remember, is a shell of the virus, so that is why my name is like that. And so again, proteins, viruses, DNA, genetic material, a capsid, and sometimes a fat envelope. If it doesn't have the fat envelope, it's called a naked virus. So naked capsid. That's what my name implies.
Great.
And then if you go there, my personal website link is right there, so you can find my email relatively easy. I'm probably the only one in the country with this first and last name, so you Google me, you'll find me.
Great. All right. Well, thank you so much for joining us today and sharing all your knowledge on viruses. This was awesome. And Erin, thank you for signing for us. Tomorrow, we're talking about sharks. That's happening at 1, and then we're talking about space on Thursday, and then microbes on Friday. Next week is all about museums. We've partnered up with the Museum of Natural History in Los Angeles, and we are psyched about that. That's going to be great. If you can support our program, please do at patreon.com/Skype-a-Scientist or paypal.me/Skype-a-Scientist. We are a 501c3 nonprofit, and so all of those are tax deductible, and we really rely on your support to keep all of this awesome science content coming to you all the time. So thank you again for both of you for joining us, and thank you all for attending and your awesome questions.
All right, thank you.
Bye.